This is Dr. Richard Fleming, and I'm coming to you today from Houston, Texas. On the 2nd of October, the plenipotentiary Dr. Peter Cutler presented some initial information to the media that I understand has raised some attention. He's done this in advance of information that I've provided him on the origins of the SARS-CoV-2 viruses, material about the eugenic genetic vaccines, the international health regulations, and a variety of other topics that he asked me to provide evidence and material that is well documented for him to present to the government of Slovakia. Since his initial presentation, there has been considerable response back, both positive and negative. But on the 10th of October, yesterday, it is my understanding that virologist Boris Klempa did an interview that Dr. Cutler asked me to respond to. I did look at that, and I'm going to share some of the pieces of that with you in just a moment. But I think it's interesting to note that while I was not there, so I don't know everything that Dr. Cutler did provide to you, and I don't speak Slovakian, so unfortunately I couldn't determine exactly what the full details of the presentation was. I have provided information that shows that Carrico and Weissman, in their 2008 paper that resulted in their receiving the Nobel Prize in Medicine last year in 2023, noted that the eugenic genetic vaccines using pseudouridine did not produce an immunologic response, but was useful for gene therapy. Now, in just a moment, I want to take a look at Dr. Klempa's responses to the journalist who raised questions about what's known as reverse transcription. So to lay out some details before playing that clip, when DNA is opened up and transcribed into RNA, that process is called transcription. It's transcribing from the language of DNA to the language of RNA. And when RNA is turned back into DNA, that is called reverse transcription. Now, I'm not certain how old Dr. Klempa is, but in the 1980s, when I was a medical student in the early 1980s, there were two pieces of very critical information that were discovered. The first was the ability of HIV to reverse transcribe its genetic code into the DNA of people. It did so quite easily in what are known as CD4 helper cells, which are critical for the immune response. And it was that mechanism that allowed it to escape being dealt with by most people who became infected and who would later die from acquired immune deficiency syndrome or AIDS. At the same time, in the early 1980s, the discovery of long interspersed nuclear elements -1 was discovered. Line 1 composes approximately 18%, or almost 1/5, that would be 20%, of human DNA. And its basic function is to take outside genetic code sequences, be they DNA viruses or RNA viruses or genetic sequences that we inject into people, which is the basic foundation of gene therapy, to take that genetic material and incorporate it into your DNA. Now, in the two presentations and the PDFs that I've provided for Dr. Cutler to present to the Slovakian government, the full details of that are laid out, not only in the presentations and the PDFs that come with it, but also the two books that are referenced, Is COVID-19 a Bioweapon and Are We the Next Endangered Species? So it's with that in mind that I want to play the next clip, because the data from the 1980s clearly showed that genetic sequences from outside of a human cell can be taken up by cells, whether it's a naturally occurring virus, whether it's a biological viral weapon that's made by people, or whether it's genetic sequences used using the technique by Carrico and Weissman from 2008, where they pointed out very clearly in the research, which I think I will now once again show you, just so you have it. So take a moment here. And we can see in this particular slide right here, this is the Carrico and Weissman slide from their 2008 publication. Incorporation of pseudouridine into mRNA yields superior non-immunologic vector with increased translational capacity and biological stability. This was the research paper that Carrico and Weissman showed that by changing uridine right here to pseudouridine, there's the carbon-nitrogen flip and the subsequent bringing in of a hydrogen atom right here to attach the nitrogen, that they produced mRNA that was now stable, would not be broken down, and they also concluded did not produce an immune response, very blunted, but would be useful for gene therapy. And also down here in slide 45, this is actually, I'll show you this. This is the Slovakian data of the variants that occurred. And this is very consistent. This is Croatia when the different vaccines were used. It did not stop the virus from coming into somebody or infecting people, which was one of the points that Dr. Klempa pointed out. He said that being vaccinated would prevent you from becoming infected. Nothing could be further from the truth. The function of a traditional vaccine is to expose you to something, an antigen, in this case a virus, so that you will build an immune response, so that when you get infected, it will shorten the amount of time to respond to it because you will have already seen it. But see, nothing in the way that vaccines work states that you won't get infected. In fact, you must get infected for any vaccine to have any benefit, to gear up your immune system again, having already been exposed to part of the virus in this case. And it doesn't prevent you from transmitting it to anybody. So for a virologist who studies viruses, and I don't know if he has any immunology basis, but for a virologist to say that vaccines prevent you from getting infected or that they will prevent you from transmitting the infection goes in the face of the science. Nothing could be further from the truth. This is the Slovakian data showing that the addition of the vaccines did not diminish the incidence of infections in Slovakia or COVID cases. It merely caused a shift in the variants, putting pressure selection on the virus, much like antibiotic resistance did. And for one of the latter statements where Dr. Klempa states that a little prick, as he put it here, would not get your ovaries or testes to produce problems for future generations, goes in the face of this slide, which comes from 2017, a research project done by Moderna using in 2017 genetic vaccines using lipid nanoparticles. This is the exact same technology used for COVID. And you can see all the organs that are infected by these lipid nanoparticle genetic vaccines, including the brain, the heart, the lymph nodes, liver, muscle, testes. They didn't have ovaries listed here, but had they checked for that, they might very well have found it. But clearly it does get to the reproductive organs, the testes in males. So that demonstrates that Dr. Klempa is not up on his science. This is 2017. Again, 1980, we knew that viruses like HIV could get into your DNA, and they did so successfully. We've talked about reverse transcriptase. There is the information out there about the SV, or simian virus 40, and the promoter genes, which I've been talking about since 2021, that can promote the production of the spike proteins. But notice, the spike proteins are an end product. It's the genetic code sequences either in the viruses themselves or the eugenic genetic vaccines are an issue. I want to take just a moment to allow you to hear Dr. Klempa in his own words as he's responding to the reporter.
Absolútne najvyššom leveli má vlastne takúto vysokú funkciu, pretože nemocný zvlády, to je ten obrovský rozdiel.
Tak uvidíme, že či to bude mať nejaký reálny dosah. Ako je možno, že aj toľko rokov po Lebo však už je to 4 roky, čo je COVID, hej, tak aj toľko rokov po očkovaní mRNA vakcínou musíme vysvetľovať, že nemení DNA.
Ja neviem, aké je to možné, to je skôr aj naozaj pre sociológa otázka. Ja len teda môžem znova opakovať, že, že mRNA vakcíny nemenia DNA, že v podstate v našich bunkách neexistujú mechanizmy, ktorými by sa to dialo.
So again, as I said, long interspersed nuclear elements, one, are the way that human DNA takes outside genetic code and brings it in to our DNA. That's almost 1/5 of our DNA that's been devoted to that, and we have known about that since 1980. And yet, Dr. Klempa, I'm not certain how old he is. I don't know if he was born in the 1980s, but I was a medical student in the 1980s dealing with HIV patients, and this data that was coming out of lung interspersed nuclear elements. So I can assure him and you that these particular genetic code sequences can and do get inserted into human DNA. And this has been known now for 44 years.
Že mRNA je molekula, ktorá je vlastne pre všetky naše bunky úplne prírodzená, pretože to je akoby ten základný recept, podľa ktorého sa v každej jednej bunke nášho tela produkujú bielkoviny. A v tomto prípade teda dokážeme tým bunkám v tom mieste v pichu lokálne podhodiť ten recept, aby začali tvoriť tie bielkoviny vírusového proteínu, čo má tu jednu veľkú výhodu, že tie bunky potom naozaj pre ten imunitný systém vyzerajú, ako keby boli infikované. A to je vlastne ten moment, kedy ten imunitný systém je naštartovaný a vlastne iniciujú sa všetky tie procesy.
And that is the beginning of the innate, INNATE, immune response, which are the T cells, which then form memory cells, which produce the chemicals that cause the inflammatory thrombotic response. And speaking to history, I am the man who published that theory at the American Heart Association in 1994, now 30 years ago.
Získavnej imunity a to nás potom vlastne chráni pred tou ďalšou infekciou v budúcnosti. Skutočne tá mRNA sa ani nemá ako dostať do jadra bunky, v ktorej je naša DNA.
So again, that is an incorrect statement. Two of them there, that getting vaccinated prevented you from getting infected. We've seen this in the United States. We've seen this around the world. People have been vaccinated. People have then been exposed to the SARS-CoV-2 virus, and they have gotten sick, in many instances so sick that they were hospitalized. And the other question here is that mRNA cannot get into the nucleus. Well, if that was the case, there would be no such thing as reverse transcription, RNA being encoded back into DNA. And the mechanism on how this gets into the nucleus is through the nuclear pore complex. This has been known for many decades, and this is the pathway in and out of the nucleus, little openings that allow the genetic code from the nucleus out into the cytoplasm or the rest of the cell. It is also the pathway in which genetic material from the outside part of the cell, called the cytoplasm, travel through the nuclear pore complex to get into the nucleus, where it can then interact with and become part of human DNA. This has been known for 40, 50 years.
A za druhé, priamo mRNA sa nemá ako integrovať do toho jadra a zároveň sa nemá ako premeniť na tú DNA.
Again, long interspersed nuclear elements 1, 18% of human DNA takes RNA from the outer part of the cell and incorporates it into human DNA. Humanity had to do nothing to make this happen. It already existed. It is part of the functionality of human cells. So to say there is no mechanism for doing this means that Dr. Klempa thinks that 18% of human DNA doesn't even exist, or that for 40 some years, 44 years, researchers who've been discussing long interspersed nuclear elements and reverse transcription don't know what they're talking about. Just Dr. Klempa does.
By mohla byť do toho jadra integrovaná. Takéto proste mechanizmy naša bunka nemá a okrem toho je to proste vec, ktorú každý by riešil ako prvú z hľadiska bezpečnosti. Takže nepochybujem o tom, že tieto analýzy už v tých predklinických fázach samozrejme boli robené. A teraz si uvedome, že zaočkovaných bolo viac ako...
So, whether that research has been done or not, for this particular virus, there are slides that I have provided showing the reverse transcription of parts of the genetic sequence from the spike protein from both the genetic vaccines and presumptively also the man-made SARS-CoV-2 viruses getting into human DNA, and it's been shown in all but 3 of the chromosomes. So he may not be aware of that research, but it's been published and it's presented in the materials I provided to Dr. Cutler.
Pol miliardy ľudí, samozrejme neviem, koľko presne z nich bolo mRNA vakcínami, ale keby sme chceli byť veľmi paranoidní, no tak tie vektorové vakcíny akože tiež vnášajú genetickú informáciu. Takže 5,5 miliardy ľudí, ako kde sú tí ľudia s tou DNA zmenenou? Kde sú? Musí byť strašne ľahké ten dôkaz vlastne získať.
Jasné, no ja vám úplne rozumiem a zaujímavé pán.
It's not easy to get the proof, but it is possible to get the proof. And I presented in the materials to Dr. Cutler evidence of that proof. And again, the proof has existed of outside genetic sequences from viruses or genetic code presented by eugenic genetic vaccines getting into human DNA since the 1980s.
Takže nakoľko vás to možno uraža alebo frustruje, lebo ja si priznám, že ja by som sa s vami chcela rozprávať aj o iných veciach ako dokola o tomto stále. Hoci je to fascinujúce, mRNA vakcíny, celá tá technológia, aj budúcnosť, ešte sa k tomu dostaneme, aby sme tu nerozprávali o prostostiach. Ale nie je to vlastne škoda, koľko energie musíme neustále venovať na dokola vysvetľovanie týchto vecí a mohli by sme sa produktívne rozprávať o niečom ďalšom. Neuraža vás to, neunavuje vás to?
Isté, že je to škoda, isté, že je to...
I think the pity here is that we do have to keep rehashing this, that he's unaware of the evidence that is presented in the scientific papers. He's unaware of the work apparently by Carrico and Weissman showing that these vaccines, these eugenic genetic vaccines, have absolutely no immunologic effect, but they are great transfection or gene therapy tools, which, by the way, if they're gene therapy tools, the very definition of what's being done in the field would be impossible if you could not somehow get the genetic sequences through the nuclear pore complex and incorporate it into the human DNA. It goes in the face of the science. And the only other thing I wanted to comment on here was his last little bit about DNA.
Ja som niekedy s nadsázkou povedal, že čo si tiež možno ľudia neuvedomujú, si to možno predstavujú ako z nejakých sci-fi filmov, že to...
So let me flip over here because I don't want to get...
Že takíto ľudia vlastne nám tu na tlačovke z pôdy parlamentu alebo z úradu vlády hovoria takéto absolútne vlastne strašidelné scenáre. Veď tí zaočkovaní ľudia samotní vlastne všetci teraz môžu byť zneistení a báť sa, čo sa stalo s jej DNA napríklad. A čo teraz, či budú mať deti a neviem čo. Takže nejde aj o to týchto ľudí ako keby trochu ukľudňovať, že teda nemusia mať obavy a ich DNA je stále tá istá. Ja som niekedy s nadsázkou povedal, že čo si tiež možno ľudia neuvedomujú, si to možno predstavujú ako z nejakých sci-fi filmov, že teda zmení sa im DNA akože v celom tele.
Nejaký Hulk, hej?
Áno, nejaký Hulk, presne. Tak som hovoril s nadsázkou, no keby sa mi náhodou v tom mieste v pichu tá DNA integrovala, no tak čo? Tak to je tu, akože to nie je vlastne...
And yet the reason for actually providing or allowing that to go through is to point out this very specific slide right here that shows that that little prick, as he puts it, that injection does get to the reproductive organs, the testes. And we know, as I've already said now repeatedly, long interspersed nuclear elements 1, if nothing else existed, would take this genetic code sequence, transfer it through the nuclear pore complex, and reverse transcribe or take RNA and put it into the DNA of human cells. By and large, I was expecting when Dr. Cutler sent this to me to actually review that there was going to be something significant to address. There wasn't. The comment has to do with whether outside genetic code sequences, whether they're coming from a eugenic genetic vaccine or viruses themselves, can get into human DNA. And we have 44 plus years worth of history from HIV and long interspersed nuclear elements 1 showing us that they do. For a virologist in 2024 to deny almost a half a century of research is to demonstrate his lack of understanding. I hope this addresses those questions. I look forward to Dr. Cutler and Prime Minister Fico actually presenting the material to the Slovakian Congress. And you're not the only ones looking at it. The Australian government now has had some of their PMs, their prime ministers, or members of parliament actually present a vote calling for a pulling of the eugenic genetic vaccines. Again, my emphasis with Dr. Kotler is one, to call for an investigation of those that funded the biological weapon known as SARS-CoV-2, which is actually, by PCR data, a combination of RS-3367, RS-SHC014, and MA15. All three viruses funded and built by the United States of America using other labs, including the Wuhan lab, and evidence of the Ukrainian lab, where the US DoD spent $419,511 on the 12th of November 2019 for a COVID report, a disease from a virus that hadn't even been reported yet to the world, but would be 7 weeks later. I hope this information has been helpful for Dr. Cutler, for Prime Minister Fico, and for the Slovakian people. Thank you very much.